Today we're going to be talking about the roles of microfilaments in cell motility. We're going to be using pizza dough to express the way the cell moves and pretzels for the microfilaments. For proteins, yellow jelly beans will represent ARP 2/3 complex, red jelly beans will represent capping proteins, purple jelly beans will be gelsolin, Smarties will be tropomodulin, fruit snacks will be filamin, and wasp will be popcorn.
During the first step, the lamellipod is pushed forward by actin monomers polymerizing onto microfilaments at the leading edge of the cell.
The new extended part of the cell creates a new adhesion site where integrins attach from the extracellular matrix proteins outside the cell. The back end of the cell is contracted forward, pulled by the new adhesion site.
Now that we've discussed the basic steps, we are now going to go over some of the proteins that make this process possible. We are now looking at a zoomed-in view of the lamellipodia.
One of the main proteins is the ARP 2/3 complex. It is responsible for making the branching pattern of the lamellipodia. WASP, represented by the popcorn, activates the ARP 2/3 complex to create a new branch from the existing microfilament.
Filament acts on the intersection of two of the microfilaments and stabilizes their interactions at the cross links.
By prohibiting the depolymerization at the minus end of the microfilament, tropomodulin prevents new free actin from being made.
When the cell wants to stop movement in one direction and start movement in another, capping proteins stop polymerization at the plus end of the actin monomers.
Gelsolin breaks the microfilament in half so they can be depolymerized faster from the minus ends of the two different pieces. They also work as a cap for the plus end of the new piece. This increases the amount of available G-actin that can be used to form microfilaments in the new direction.
Although we touched on several of the key proteins, microfilaments are very complex and require these and many others to successfully polymerize, depolymerize, and stabilize actin.